Hi and welcome back. Let me introduce you to Asha. She lives with her mom, dad and 2 brothers in the West Indies. Unlike the rest of her immediate family, she loves to eat karaili, also known as bitter melon. The only time she gets to eat it is when she visits her grandmother, who shares her love for this food. She spends Saturdays with her grandmother, where they regularly enjoy a meal of curried karaili and an ice-cold glass of morbi. Her brothers constantly tease her about her karaili mouth. They can't understand how she could love it so much. One day during biology class, her teacher introduced them to genetic inheritance by doing an experiment with them. They did a taste test using strips of filter paper soaked in propyl thiouracil, also known as prop. Everyone except herself and her best friend Savi said that the paper tasted bitter. Her teacher then explained that prop is closely related to phenylthiocarbamide, PTC. She told them that the ability to taste PTC and similar chemicals has a genetic basis. That is, their genes determine whether they can taste it or not. Her teacher also explained that the ability to taste it is the dominant trait. This was reflected in their experimental results, since all but 2 students were able to taste prop. On learning this, Asha started to wonder, could that be why she loves karaili, even though her family finds it horribly bitter? But if so, from whom did she inherit the inability to taste PTC? Maybe her grandmother? But how would that work if neither of her parents seemed to have this trait? Are you also wondering? Or do you already have an idea? By the end of this episode, you will be able to answer all of Asha's questions and more using the monohybrid cross for the F1 and F2 generations. So let's get started with a very important question. What is a monohybrid cross? Well, this is a type of genetic cross. A genetic cross is an arranged mating of selected individuals with the goal of mixing desired genes. Now, a monohybrid cross selects and mates individuals that differ in just one characteristic, and that therefore leads to a mixing of versions of one gene. Sensitivity to PTC is mostly controlled by one gene. The monohybrid cross is based on experiments conducted by Gregor Mendel in the 1800s. During his decade-long investigation, he collected data from over 30,000 garden pea plants. Analysis of this data led to the development of the basic rules of genetic inheritance. The inheritance of characteristics that are each controlled by one gene is referred to as Mendelian inheritance. You can predict the results of a monohybrid cross using a genetic diagram, and that is what I will be demonstrating to you in this video. Before we begin the diagram, let's establish what we already know. 1. Human cells are diploid. This means that each family member's cells will have 2 copies of the gene that controls PTC sensitivity. 2. The ability versus inability to taste PTC are controlled by 2 different versions of the gene called alleles. 3. The ability to taste PTC is dominant and the inability to taste PTC is recessive. So let's start by defining our symbols. We can use the letter B for bitter because its capital and common forms are distinct from each other, reducing the chance of confusion as we go along. Now let's start with Asher's grandparents. This would be the P0 generation. In Mendel's experiments, the P0 generation was a cross of true breeding individuals with contrasting traits. This means that the parents had homozygous genotypes for their respective traits. We already know that her grandmother loves Kareili, so let's assume that means that she cannot taste PTC. Asher's deceased grandfather hated Kareili, so let's assume that he was able to taste PTC. Inability to taste PTC is recessive, so only the homozygous genotype would allow this trait to be expressed. Therefore, the genotype for Asher's grandmother can only be homozygous recessive. Ability to taste PTC is a dominant trait, so Asher's grandfather can have either a homozygous dominant or heterozygous genotype. As we are following the pattern of Mendel's experiments, we will assume that his genotype is homozygous dominant. Now we have to figure out the distribution of alleles in their gametes. And to do that, we need to take a deeper look at their chromosomes. Being diploid means that their DNA is organized into pairs of homologous chromosomes. Each pair carries the same set of genes in the same locations, although the genes aren't always identical. Gametes are produced via meiosis. During meiosis, each homologous pair is split up. Each member of the pair ends up in a separate gamete. And this is what we demonstrate in the genetic diagram. So, returning to our diagram, let's take a look at the genotypes. Since the alleles are identical for each grandparent, there is only one type of allele that can be found in their gametes. The gametes from their grandmother will all carry the allele for inability to taste PTC. The gametes from her grandfather will all carry the allele for ability to taste PTC. And now let's see what combinations fertilization can produce. Here we use a Punnett square, which is essentially a grid. It helps us to figure out possible combinations in an organized way. The gamete of parent 1 is placed on the topmost row, and the gamete of parent 2 is placed in the leftmost column. Then we combine. Since there is only one allele per parent, only one combination can be produced. This new combination of alleles is called heterozygous because each member of the pair is different. This is the F1 generation, the offspring of a cross of two pure breeding parents, as per Mendel's experiment. The genotypic ratio is 100% heterozygous. The phenotypic ratio is 100% of the dominant trait. In this case, it means all the offspring are able to taste PTC. This is the generation that Asher's parents would belong to. But of course we do not stop there. Asher and her siblings would belong to the F2 generation, and we haven't gotten there as yet. In Mendel's experiment, he allowed individuals of the F1 generation to then self-fertilize. This is equivalent to a cross of parents with heterozygous genotypes. We already know that Asher's parents both hate Kareili, which could relate to the ability to taste PTC. In this case, we will assume that they are both heterozygous, instead of homozygous dominant. This time, the distribution of alleles in the gametes is slightly more complicated. Let's return to our chromosomes. As before, the alleles are found on homologous chromosomes, which are then split up and sent to separate gametes. However, in this case, since the alleles in the homologous pair were different, the gametes will have a 50-50 split. Half will carry one allele, and half will carry the other allele. So let's write that into our genetic diagram. Each parent, therefore, produces two types of gametes, half with one allele and half with the other allele. So, during the fertilization phase, what are the possible combinations? To figure that out, let's once again use the Punnett square. As before, the gametes for parent 1 go on the topmost row, and the gametes for parent 2 go in the leftmost column. This time, each parent has two types of gametes. Both are therefore added to the grid. And these are the possible combinations. This is the F2 generation, the offspring of a cross of two hybrids, as per Mendel's experiment. The genotypic ratio is as follows: 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Since the heterozygous genotypes would express the dominant allele, the phenotypic ratio would be the sum of the homozygous dominant and the heterozygous genotype, 1 plus 2. So the phenotypic ratio would be 3 dominant to 1 recessive. In this case, it would be 3 able to taste PTC to 1 unable to taste PTC. And hey, that's Asher. This is also known as a Mendelian ratio for an F2 generation. So now we have seen how Asher could inherit the inability to taste PTC, even though both her parents have the opposite condition. The inability to taste PTC is a recessive condition which can be completely hidden in one generation, then reappear in another generation under the right circumstances. And that is what Mendel's experiments demonstrated, and his work thus became the foundation of our understanding of genetic inheritance. I hope this was of help to you. Now, of course, genes are not transferred from parent to offspring one at a time. In a future video, we will explore the dihybrid cross and the even greater combinations it produces. And we will also explore genetic inheritance that does not give us Mendelian ratios and why. So make sure to subscribe as we continue this series in the following days and weeks. Till then,